Okay, so in this video we're going to talk about the liver, the gallbladder, the pancreas, and the large intestine. Earlier we talked about how the small intestine had three parts, like the duodenum, the jejunum, and the ileum. And there was this kind of important area right where the duodenum forms a bend, and that was the place where the liver associates with the pancreas, and they both drain their secretions through the same duct into the duodenum. And so what we'll do is really focus in a more detail on the functions of your liver and the structure of your liver, as well as the structure and functions of your pancreas and how they associate to help secrete digestive juices. So we've talked about the liver as being an accessory digestive organ because the liver's main function isn't necessarily digestion, right? Now it does process some of the nutrients that you absorb from your digestive tract, but it has a lot of different functions too. Like it makes plasma proteins like albumin and complement. It's involved with storing certain vitamins as well. It's also involved with helping to get rid of damaged red blood cells. And what's also pretty cool too, guys, is that under really extreme conditions, your liver can actually make new red blood cells. So the liver has some erythropoiesis ability as well. So like if your red blood cell count's low, like if you're having blood drawn or something and you start losing a lot of blood, your liver can help make new red blood cells, which is kind of cool. That's not its main function though, it's sort of a side function. So, but in terms of the digestive system in here, we'll talk about the liver's digestive function and really focus on that, right? Now we'll be coming back to the liver a lot as we go through the different organ systems because it comes up again in terms of its interrelationship of like blood pressure and immune defense and that kind of stuff. So with the liver, its only digestive function is to produce bile, right? If digestion is the breakdown of things, the way the liver breaks down nutrients in your digestive tract is it produces bile. Bile is an emulsifier. So here's the thing that's kind of weird though about bile, is it doesn't break chemical bonds in fat molecules. All emulsifiers do is separate fat into smaller pieces, right? So let's say if you have a big old fat globule in your duodenum, what bile does as an emulsifier is it would break apart that fat globule into smaller, more digestible pieces, and that increases the surface area for digestion. Now you're not actually breaking apart the fat molecules, you're just sort of splitting them apart rather, okay? So you store excess bile in the gallbladder, right? So it's a bladder of bile, and its chief function is just bile storage. Now the gallbladder is under the influence of certain hormones like cholecystokinin, and it also receives vagus nerve input, so that while you're undergoing digestion and those hormones are being released and you have nervous impulses going to your gallbladder, the gallbladder has smooth muscle in its walls that can contract and force that bile out into your duodenum, right? So if we look at this picture again, you guys have seen this one before. We see here is the liver, and the liver is actually pretty big. I want to say it's larger than the football. And the largest part of the liver is the right lobe here, and the smaller part is the left lobe, and they're separated here by this falciform ligament. Now, the right lobe of the liver has a lot of cells, a lot of blood flow, and we'll talk about the structure histologically here in a minute. But essentially all the bile that's made by your right lobe of your liver is drained by this right hepatic duct, okay? And then all the bile that's made by the left lobe of your liver is drained by this left hepatic duct. Then they come together and form this common hepatic duct. Now, the common hepatic duct will lead into the common bile duct after it passes this duct here called the cystic duct, and it drains that bile towards the duodenum. But it doesn't always release there because what happens is we have a sphincter there that controls the release of bile and pancreatic juice. We call that the hepatopancreatic sphincter, or sphincter of Oddi. Okay, so when bile drains down here, or not drains rather, but it kind of falls down there, it will actually backfill up back towards the gallbladder, right? And so then the gallbladder can store that excess bile here. Now, we also see that this is associated with the pancreas, and the pancreas is this sort of fish-shaped organ here. You have a head, a body, and a tail, and it has a main pancreatic duct that helps drain the pancreatic juices made by the acini into the same space. Now, this shared space where bile and pancreatic juices meet is the hepatopancreatic ampulla, or the ampulla of Vater, okay? So that way bile juices and pancreatic juices are co-released into the duodenum. And if you guys remember, the duodenum is going to receive acidic chyme from the stomach. But the problem is that your duodenum is not adapted to low pH conditions. So what we have then is that bile juices and pancreatic juices are going to be slightly basic, and what they do is kind of help to neutralize the acidity of your chyme, and that way pancreatic enzymes can function more effectively. So the enzymes that are produced by your pancreas only function at a certain pH range, and they're not going to work if your chyme is too acidic. All right, so if we look at the histology of the liver, and you took a cross-section through it, you'd find all of these sort of hexagonal-shaped structures here. Okay. Now, this hexagonal-shaped structure is separated by these septa, so you have connective tissue. If you look closely, there's a central vein here where all the blood is drained back towards your heart. Okay. And so if we zoom in on this central vein, and we check out this picture here, you can see there's a lot more detail here. Now, we call this all the sinusoids, okay? And so if you look closely here, you guys, this vein down here is the portal vein. And if y'all remember, the portal vein comes from the veins that drain your digestive organs. So we said that the veins from like your small intestine and your large intestine and your spleen and your stomach and your esophagus, those all drain together into the portal vein. Now, once the portal vein enters the liver, it then divides into smaller veins that then lead into these sinusoids. Okay? So what would be in this portal venous blood? Well, it would be nutrient-rich blood because it's coming from your digestive tract, but it would be oxygen poor, right? So it'd have low oxygen, but nutrient-rich blood. And it hasn't been processed yet because it hasn't gotten to the liver yet. This is just like newly absorbed nutrients that came from your digestive tract that are from the portal vein. Now, if you look closely, this portal vein kind of divides and it goes on in different sections. And if you guys see here, that leads into basically these plates of hepatocytes. So all of these cells here are the hepatocytes, and you'll see that they actually line the spaces by these veins or sinusoids. Now, what this red thing over here, though, is actually the arterial blood that your liver receives. So if the liver only received that portal venous blood, it would be pretty oxygen poor, right? Because your digestive organs have already used the oxygen there, although there would be a lot of nutrients, but it would be low in oxygen, so it would be hypoxic, right? Now, what your liver also receives then too is arterial blood from the aorta, and you can see that here are the arteries, but the arterial blood mixes with the venous blood. So now you have like a mixture of oxygen-rich and nutrient-rich blood going through these sinusoids. And so as this blood passes through these spaces, what happens is all these hepatocytes start processing it, right? So it's almost like this is the factory of your liver, right? So we have nutrient-rich blood that's coming down the line, and these hepatocytes are basically taking nutrients out of the blood. They're processing it, packaging it, storing it, or repackaging it and sending it along to other organs in your body, okay? So each one of these cells is a hepatocyte, right? And so when you think about the functions of hepatocytes like storing glycogen, so you can take glucose out of that blood and store it in the form of glycogen for later use. And that's what glucagon act on, right? Remember we said glucagon helps to increase your blood sugar? Well, it causes your hepatocytes to release some of their stored nutrients. These hepatocytes also process lipids, and they package those lipids into lipoproteins that are sent along to your body cells, okay? These hepatocytes can also interconvert nutrient types. Like you can convert sugar to cholesterol, or you can convert sugar to fat, right? This is done by your liver, so it's pretty cool. Now, what's this green stuff, though, next to these cells? Well, those are bile ducts, okay? So what we'll focus on here, though, with respect to digestion, is that next to all these hepatocytes, there's these tiny, tiny little bile ducts called bile canaliculi, okay? So the bile canaliculi, they collect the bile that's secreted by these hepatocytes, and then all these bile canaliculi come together to form these larger bile collecting vessels. And these larger bile collecting vessels here converge into the larger bile ducts that we saw in the previous slide. So remember how we said that the right hepatic duct drained all the bile from the right lobe of your liver? We're saying that that right hepatic duct would actually receive all the bile from all the bile canaliculi from every single hepatocyte in the right lobe of your liver. Okay, so it's actually pretty cool. That's where it all comes from. So this would actually go back down out of your liver. And so where does the blood flow then after it enters the liver? Okay, well, we said that arterial blood here mixes with the nutrient-rich venous blood from your digestive organs, and where it mixes, it goes past the hepatocytes and eventually leads into the central vein. Now the central vein here drains up towards these interlobar veins and then eventually through what's called the hepatic veins that then drain into your inferior vena cava, which goes right back to your heart. Okay, so what you're seeing here then is that the liver gets a lot of blood flow, right? Because is this the whole liver right here? No, exactly. In fact, if we back up, that's just this little piece we're looking at. In fact, if you look even zoomed out more, that's just that one little piece. And if you zoom out to the whole liver, wow, that's just a small section of the entire liver. So there's a lot of these sinusoids, right? And what this means then is that there's a lot of backup. Like if you damage a piece of your liver, well, there's a lot of areas that have the same function that might be just as healthy. Okay. What's also cool too, you guys, is inside the liver you have stem cells that can regenerate damaged or lost liver tissue. That's also really cool. So unlike other organs, the liver has high regenerative capacity. Like if you have liver damage, it's going to recuperate better than, let's say, if you had like heart muscle damage from a heart attack or something. Okay. So remember, the hepatocytes, they're adapted cells, and they're involved with processing. So they have a tremendous amount of smooth and rough ER. If you guys remember going back to general biology, we talked about how smooth ER was involved not just with synthesizing lipids, but it also helps to detoxify drugs and toxins in your body, right? So smooth ER is involved with breakdown. And that kind of makes sense with respect to the liver, because if the liver is involved with detoxification, it would make sense that the cells of your liver have more of the organelle that detoxifies, which is smooth ER. So if you could take out an individual liver cell and look at it, it would have a way more smooth ER than like other cells of your body. So it's really good at detoxifying. That actually makes sense, right? And so as far as your body's concerned, you guys, like all toxins are the same, whether it's a drug you buy over the counter, or if it's a street drug, or if it's something that maybe you've ingested in your own food, right? If it's a chemical that your body doesn't normally use and it's toxic, your liver processes that, okay? Rough ER, if you all remember, is involved with creating secreted protein. So it makes sense then that the liver has a lot of rough ER because the liver also secretes a tremendous amount of protein. Most of these proteins the liver secretes end up in your blood plasma, and the function of that is, for one, you have albumin, right? So albumin is involved with like maintaining proper osmotic balance in your blood. It also transports certain hormones, and your liver also makes complement proteins, which are involved with immune defense. So these proteins that are made by your liver have a pretty wide variety of functions, which is pretty cool. So it processes bloodborne nutrients, stores fat-soluble vitamins, so like D, E, A, and K, performs detoxification. Wait, okay, let's back up, you guys. Detoxification, what's the organelle that the liver would use to detoxify? Which organelle would we use? The smooth ER, exactly, good, right? So you have a lot more smooth ER there. And so when you talk about drug tolerance, what do you guys think is going on there? Like if someone can tolerate a drug better. Yeah, their body can process it easier, right? So where do you think that processing is occurring? Smooth ER in the liver, you got it. So when you hear about like people who are drug addicts and they can like take enough heroin to kill an elephant, but they're fine, you know what I mean? The difference between them and people who would die from that dose of heroin would essentially be, that they have way more smooth ER and they're able to process those nutrients faster, such that they don't actually die. So that's kind of cool. So how does that explain then where if people, let's say drug users, if they go clean, but then they go back to using drugs again, right? Let's say if they go clean for a little bit, what's going to happen to the smooth ER and the hepatocytes of the liver? Is it going to stay like abundant? No, it's going to atrophy back to like probably like a normal level, right? But then let's say that drug user then goes back to using drugs. They're probably going to take the dose that they remember taking, right? But is their body adapted for that anymore? Nope. That's why they usually overdose. So when you hear about people, like these sad stories where people say, oh, yeah, they were clean for like 6 months and then they, went back to taking drugs, that's typically when they'll overdose because they think that they need more, even though their body's not adapted for that anymore. Yeah, there's those cases all over the place. So what's kind of cool to you guys, you produce 900 milliliters of bile every day. So that's almost an entire liter. Like here's a water bottle, right? This is a 1 liter water bottle. 900 milliliters is 9/10 of that. So that's how much bile you can produce in a day, which is a tremendous amount, right? So it makes sense you want to have a bladder to store that. You're releasing it when you eat, and some of that ends up in your feces, right? But a pretty large majority of it is actually reabsorbed back into your bloodstream and then sent back to your liver and reused, which is pretty cool. So you can recycle some of the components in bile again. So when you think about bile, you guys, it looks kind of yellow-green. It's alkaline, so it's kind of basic, right? It's yellow-green because of the bilirubin that's in bile, and that's actually what gives it its name bile. Now, bilirubin is the breakdown product of hemoglobin, and hemoglobin is something you find in your red blood cells. So what's kind of weird is that your liver uses damaged red blood cells to essentially make bile through the breakdown of hemoglobin. Okay. Now, these bile salts are cholesterol derivatives, and they're involved with fat emulsification. And then bilirubin is the pigment that you find in heme. Remember, heme is in hemoglobin. And so bilirubin has more of a yellowish color, right? Let's think about this, you guys. If your liver normally removes damaged red blood cells from your bloodstream and puts that into bile, what happens if your liver is failing? What starts to accumulate inside your bloodstream? Damaged red blood cells, which will spontaneously break down into bilirubin. That bilirubin then, instead of being sent into bile, because your liver can't do that anymore, it starts depositing in your tissues, and that makes your skin kind of yellow, right? That's what jaundice is. So you're saying that instead of incorporating bilirubin into bile, it's now just ending up in your tissues. That's why people have that sort of jaundice appearance. Now, it's not always prominent on skin, especially because just based on varying skin tones, but usually you can see jaundice really well on the sclera of the eyeball, right? So normally your sclera should be fairly white. Now, it's going to be pretty prominent there because even bilirubin is going to accumulate on the sclera, so it's going to make you look like you have yellow eyes, right? So it's kind of interesting. Okay, so what's also interesting here too, guys, is that once bilirubin ends up in your digestive tract and it's broken down by bacteria, it ends up into something called stercobilin, which is a brownish color. So the brownish color of feces is due to the breakdown of the bilirubin by the bacteria in your gut. That's why poop is brown, right? So, but is poop always brown? No, absolutely not, right? It can be anywhere on a gradient of gray, which I've never seen before, but apparently gray, to brown or green. Okay. Now, if poop is green, it's just way too much bile in there. In fact, you've had a lot of bile, so much so that the bacteria in your gut couldn't break it all down. So when it comes out, there's still some bile in there. So you might wonder, well, how do you end up with green poop? Well, really fatty meals. So if you eat a really fatty meal and you release a tremendous amount of bile, such that your bacteria don't break it into stercobilin, then it's going to stay green like bile, which makes sense, right? If it's brown, then essentially you've released, just enough bile, and your bacteria process it, which makes sense. Cool. What about the gray thing? Feces can be kind of grayish, whitish. Do you guys know that? That happens when you don't release any bile. So let's say if you had some sort of bile duct obstruction or some dysfunction of your liver where you stop producing and secreting bile, then you never incorporate that in with the digestive contents, and so they stay kind of bleached from the acid and enzymes, and so they come out as sort of a grayish, whitish color. So if you ever see that and you have like really light feces, not you, but let's say a patient, that could be you too. But that's a bile problem. Like you're not secreting enough bile, which makes sense. Cool. All right, guys. So we have cholesterol that's incorporated into bile. In fact, excess cholesterol is incorporated into bile. This is how you get rid of excess cholesterol out of your bloodstream. Triglycerides, phospholipids, and electrolytes. All that's in with these, bile solution here, bile juice. Okay. Now you can reabsorb some of this stuff too. Like you can reabsorb some of those electrolytes. You can reabsorb the water, the bilirubin, even itself. You can reabsorb. So oftentimes this bile is recycled. So moving on to talk about the pancreas, we talk about this associated with the liver because they share the same exit point, or ducts, right? And so we know that the pancreas has an endocrine function, which comes from the pancreatic islets. If y'all remember, the islets make up about 1% of your pancreatic cells, and they secrete hormones like insulin and glucagon, okay? Now that's important to note, because if you have pancreatitis, it's not going to only affect digestion, but it's also going to affect endocrine function, okay? Now the exocrine function of your pancreas is the digestive function, right? And this comes from the pancreatic acini. Acinus means grape. So an acini is a grape-like cluster. And so what you find then is that these acini are clusters of secretory cells that surround a duct, and they secrete pancreatic juices into these ducts that converge into a main pancreatic duct that moves on over towards the hepatopancreatic ampulla, or the ampulla of Vader, right? So what's kind of cool too, guys, is that when you secrete pancreatic enzymes, they're actually in their inactive form, which we call zymogens, okay? And it makes sense because the pancreatic enzymes you secrete digest lipids, proteins, carbohydrates, and nucleic acids. What's your body made of? Lipids, proteins, carbohydrates, and nucleic acids, right? So if you can make enzymes that can digest the molecules that your body's made of, you want to be really careful with these enzymes, right? In fact, you don't want to release them in their active form because they can start to auto-digest the pancreas, and that can happen with pancreatitis. That's the one thing that can really cause this to spiral out of control. But normally, you secrete them in their inactive form. And then, once they get over to the small intestine, the duodenum, there are other enzymes along the brush border of the duodenum that can activate those enzymes. That way, they're only active outside of your body, inside the duodenum, right? Bless you, not inside the pancreas. That's important to note. Okay, so if we look at the pancreatic acini here, you guys, this is zoomed in here. And so remember, the enzymes that your pancreas is secreting, those are protein. So what organelle would you want to have a lot of if your pancreatic acini are secreting protein? Do you guys remember? Like which organelles make protein? Rough ER, good. And just ribosomes, right? But the rough ER is rough because of the ribosomes, okay? Cool, very good. And you also want to have these nuclei that are active and can basically help make, transcripts, mRNA transcripts that can make protein. And you have a lot of mitochondria here too, right? Because it takes a lot of energy to make that protein and keep these cells active. So if you guys look here, these enzymes are packaged into vesicles. And these vesicles are shown here in kind of dark color. Now, these vesicles are full of the zymogen granules, which are essentially the proenzymes or the inactive enzymes. Then they're secreted by exocytosis into this duct. And even once they're in that duct, they're still not active, right? But then what you do have here is sort of an inactive pancreatic juice, which is full of bicarbonate and water and proenzymes, okay? Then that juice makes it on over to these ducts that converge into the main pancreatic duct, and then they're eventually released into the duodenum, okay? Now you make 1.2 to 1.5 liters of pancreatic juice every day. Here's my water bottle again, you guys. This is a 1 liter water bottle. So you're making 1 and 20% of this or 1.5 of this in pancreatic juice every day. That's a lot of juice, right, from your pancreas. In fact, your pancreas isn't even nearly as close to a liter in volume itself. So it's making way more than its own volume in juices every day, which is kind of amazing. So what is pancreatic juice made of, you might say? Well, it's a lot of water, a lot of bicarbonate, which is a base, right? You got some electrolytes, and a whole lot of protein. But the protein that you find in pancreatic juice is digestive enzymes, right? So those digestive enzymes like proteases, lipases, you have nucleases, which break down, nucleic acids. And so there's a lot of different types of enzymes here. Now, they're secreted in their active form, but they're not activated until they achieve sort of the proper balance of electrolytes from bile. And so, although they can be active, unless they mix with bile, that's when they're really actually active. Okay, cool. Now, the proteases are secreted in their inactive form, but they're cleaved by the enzymes that are along your brush border of your duodenum, and that's when they're actually activated in the duodenum. So that's kind of important to know. So if you guys look here, we can see our stomach. This is kind of a weird view, huh? Here's our stomach. there's our pyloric sphincter. Here's the duodenum, and so this view would be kind of like you're like in the duodenum, looking up into it, right? Like, okay, what's going on in that duodenum there? Okay, so you can see where they cut away a piece of the duodenum. Okay, you can see where the bile juices and pancreatic juices are co-released into the duodenum, and you're seeing that these enzymes like trypsinogen, chymotrypsinogen, and procarboxypeptidase, those are in their proenzyme inactive state. Okay. In fact, remember how we talked about the ogen, like with pepsinogen? That's with its inactive form, right? Now, these are actually activated by these membrane-bound enteropeptidases, which actually cleave those enzymes and they get them to be active, right? It's kind of like, you guys ever like get a new electronic device? You got to pull out that little tab. You pull the tab out of the battery pack, and like that's what gets it started. I don't know where the heck I've ever seen that before. You know, maybe like a watch or something, like a really cheap watch. Or no, you know what it is? It's like Halloween lights and stuff. You know, you might pull the tab out of the thing, and that's what gets the battery to make contact. Then you can actually turn the thing on, and then you have like a little, jack-o'-lantern thing. So imagine these membrane-bound enteropeptidases are kind of like pulling the tab out of those enzymes, right? They're removing the thing that allows those enzymes to now turn on. Now you get these, these enzymes that can start to chew up things. So these are all peptidases, right? Which are proteases, rather, which essentially break peptide bonds between protein. Now the reason why you have 3 different types of proteases, you guys, is they cut in different ways. Like the procarboxypeptidase only cuts from one end, right? And there's other enzymes that only cut from the other. So you're chewing the proteins from both sides, and there's even enzymes here that actually cut the proteins down the middle, right? So you're chopping up the middle, and you're also chopping up on the sides. And so using all 3, you're really digesting that protein quickly, right? Because if you only did it one way, it would take you forever to get along the protein, right? If you're only eating one side, okay, but you can eat in many different ways, which are what these proteins or these enzymes are doing. So in their active form, we have things like trypsin, chymotrypsin, and carboxypeptidase. But remember, you guys, these are only going to function at a certain pH range and a certain electrolyte balance, which is mediated by the presence of bile and the pH that's in your duodenum. By the time you get on over to like other areas of your small intestine and even your large intestine, these enzymes are no longer active. They've done their job, and they've kind of, you might say, burned out, but essentially what happens is that in different pH and different circumstances, they don't work anymore. They only work at very specific ways or circumstances. Okay, so what this slide is showing you guys is the process by which pancreatic juices and bile juices are stimulated to be released. So going back to the 3 phases of gastric secretion. Do you guys remember we had the cephalic phase, the gastric phase, and the intestinal phase? The cephalic phase is when you're thinking about food, and the vagus nerve input from your brain can start to get like your stomach excited. Now your vagus nerves also go to your they also go to your gallbladder, and they can very gently kind of cause the gallbladder to contract. So you can get a little bit of bile to be released into the duodenum during the cephalic phase. So just thinking about food can get a little squeeze of bile into your duodenum. Not a lot. The vagus nerves kind of have a weak effect on the gallbladder, but they can get a little bit of bile out. Okay. Now you might wonder, well, how do you get the most bile out? Well, it's through the release of those enterogastrones. So if you guys remember going back to the 3rd and final phase of gastric secretion, we talked about the enterogastrones that are released by the small intestine, right? So let's look at that here. So if y'all remember the stomach released chyme into the duodenum, then we entered the intestinal phase, right? Now briefly, in the first part of the intestinal phase, you saw a rise in gastrin, and that allowed for more chyme to enter. But then the major part of the intestinal phase was the release of inhibitory enterogastrones that inhibit the stomach. So if y'all remember, those enterogastrones were things like cholecystokinin, which is CCK, secretin, or vasoactive intestinal peptide, right? Now all 3 of those hormones that are released by your duodenum via the presence of chyme, they actually inhibit your stomach, right? But, this is the big but here, right? Is that they also stimulate your gallbladder and your pancreas to secrete their secretions in the duodenum. So although these hormones are inhibitory on the stomach, they're also excitatory on other organs. So the name cholecystokinin, CCK, chola is the root for bile. And so cysto is also the root for gallbladder. So cholecystokinin stimulates the gallbladder to contract. So once the chyme enters your duodenum releases a hormone that effectively tells your gallbladder to start squeezing in bile, which actually makes a lot of sense, right? Because you want to have the bile in there when the chyme's there. Okay, good. That's what's shown in red here, you guys. So if you follow this out, you can see the little red molecules floating around your bloodstream, and eventually they make it on over to the liver and gallbladder where they can stimulate the synthesis of more bile and the secretion of bile. Good. Now, cholecystokinin also has a small effect on your pancreas because it causes your pancreas to secrete pancreatic juices, right around the same time as it's stimulating your liver and gallbladder. Okay, so the same hormone that stimulates your liver and gallbladder also stimulates the pancreas. It has an effect on this hepatopancreatic sphincter as well, you guys, because it causes the sphincter to relax. That way you can actually get these juices to enter the duodenum. Okay, but what about secretin, which is shown in yellow here? Well, secretin is a hormone that can circulate around your bloodstream. It also stimulates the pancreas to secrete juices, but secretin also stimulates the release of insulin. So how would it make sense to release insulin right around the same time chyme is in your duodenum? What do you guys think? Think about the effect of insulin. What's the effect of insulin? It lowers blood sugar. So why would you want to release the hormone that lowers your blood sugar once chyme is in the duodenum? What's going to happen next when that chyme's in the duodenum to your blood sugar? It should rise, right? Because you're going to start to absorb that sugar. Okay. So let's think about this. If the chyme's in the duodenum and your blood sugar would spike all of a sudden, what you can do is release secretin that stimulates your pancreas to release insulin. That way, once you start absorbing that sugar, the insulin's already there. And that way you can actually keep your blood sugar from getting too high, and you're helping to transport that sugar into your body's cells. So that makes sense. It's just timing, guys. Like the way I look at this is kind of like an orchestra of events, right? Like all the right players have to be playing the right thing at the right time to make this like orchestra of digestion, which sounds disgusting, but it is what it is. All right, cool. Now, secretin also stimulates the liver, you guys, to produce bile more rapidly. So it's kind of cool. All right. So now in terms of motility of the small intestine, you guys, it's actually mediated by reflexes. So we've talked a little bit about the motility of like your esophagus and your stomach because those were under control of like local reflexes for your esophagus. And for your stomach, it was under the control of things like gastrin, local reflexes as well due to stretch, as well as the vagus nerves can all get your stomach to move around. But what about the small intestine, right? How do digestive contents move along the small intestine? And like your small intestine is not really always that active, right? So it has to be conditionally active. Well, what we have here, you guys, is that when chyme enters the large intestine, what happens then is that the ileocecal sphincter relaxes. Okay, so this ileocecal sphincter is a sphincter that separates your small intestine and your large intestine. In order to get that chyme to enter the large intestine, that sphincter has to relax. Okay. Now, what causes this to relax is something called the gastroileal reflex. So once you eat some food and it reaches your stomach, the nerves of your stomach can communicate with the ileocecal sphincter, and so that once you eat the food, then your ileocecal sphincter relaxes. So here's the thing. When you have digestive contents that are near your large intestine, those are not the digestive contents you just ate, right? That would be like the last meal you ate. So I'll give you guys an example. Okay, let's say you just ate breakfast, right? And that breakfast went down and hit your stomach. You just stimulated a gastroileal reflex. But the chyme that enters your large intestine from your small intestine at that moment isn't your breakfast. It's the dinner you ate the night before. Because it took that long for the dinner to get through your small intestine, right? And then once it gets right by the large intestine, your next meal stimulates the movement then of dinner into your cecum, even though you just ate breakfast, right? So it's just sort of letting the next stuff through. So you're making room, right? You're kind of like, all right, out with the old, in with the new, right? Okay, time to move on, right? We've done our job, you've digested, and time to go to the large intestine, where that's your next fate, right? So that's that gastroileal reflex. Now, gastrin also increases the motility of your ileum, right? So once you reach the gastric phase and you start releasing a bunch of gastrin, your ileum also start moving a lot. And it's going to move those previous digestive contents over to the large intestine. Again, you're just making room for the next meal. So that actually makes a lot of sense. Now this ileocecal valve closes, and it's a one-way valve once the chyme enters the cecum. That way it can't flow back to where it came from. It's one way, from small intestine to large intestine or cecum, and then you won't go back again. So it prevents regurgitation in the ileum, and that will be a good thing. If you have regurgitation in the ileum, you're saying that feces are basically coming back in your small intestine. You know what I mean? And that wouldn't be good because there's a lot of bacteria there, and you don't want to have congestion of your small intestine. Okay, so let's check out the anatomy of your large intestine really quick. First of all, what would be here that's missing? Do you guys know? Small intestine, exactly. Good. And so all of this, by the way, in yellow is the mesentery proper. We talked about how mesentery proper anchors your small intestine as well as parts of your large intestine. Now, do you guys see this little piece right here? That's one little piece of the distal ileum that they left, just to show that when you cut off the small intestine, okay, there's a little piece right here that connects with the cecum. Okay, so this is the ileocecal valve or sphincter, so that when digestive contents and chyme go through this valve, through the gastroileal reflex, they enter the cecum. And the cecum is the beginning of your large intestine. Okay. Now the cecum also has your vermiform appendix attached. Now the appendix is a tube that's continuous with the cecum, and what's in the appendix is essentially good bacteria, right? So what's a good bacterium? Well, good bacteria would be the bacteria that are in your large intestine that you use to either outcompete the pathogenic bacteria like C. diff, right? Or they're bacteria that make things that your body uses, like certain vitamins, like B vitamins and vitamin K, are actually synthesized by some of the bacteria in your gut. So it's kind of cool. So those would be good bacteria. So the way I look at these guys is that the appendix is kind of like the nuclear bunker of your large intestine, right? Because it's like a place where good bacteria can hide. Because if things go bad in the large intestine and the whole population of bacteria is destroyed here in your large intestine, you can repopulate the earth, which is your large intestine, because the bacteria can kind of come out of the cecum and go, what happened? What happened? And they can repopulate, the large intestine. Okay. Good. So the cecum leads into the ascending colon up here, you guys. This is the ascending colon. This is the right colic or hepatic flexure. Then it goes to the transverse colon. Then we have the left colic or splenic flexure, to the descending colon, to the sigmoid colon, and then the rectum, and then anus. So you guys can see the anal sphincters here, which we'll talk about here in a minute. Okay, so you guys see how there's like lots of pouches that are along the length of the large intestine? Those are called haustra. So the haustra pouches, right? That's the way I think about it. So the haustrum here, they house feces, right? I think that's why maybe in my mind why they're called haustra. Okay, and so when the large intestine contracts, you move feces from one haustrum to the next haustrum, to the next haustrum, right? In fact, you guys ever seen really constipated feces? They kind of have the appearance of haustrum. Lumpy, very lumpy, right? Kind of makes sense, yeah. And it makes sense when they're dehydrated because they take on the shape of where they're dehydrated. If they're not dehydrated, they don't take on that shape. So it's kind of interesting. There's also these little things that hang off the large intestine here, you guys. They're called epiploic appendages. And the epiploic appendages are like the bling of your large intestine. But they're basically like pieces of adipose. It's just more place to store fat, right? So let's move on to talk about the anus. Why not, since we're here? So if we look at the anal canal here, you guys, what's really cool in the rectum is we actually have these rectal valves or flaps. And these rectal valves ensure that like feces don't just leak out of your rectum through your anus, right? That's pretty good. Okay, so you have these little one-way rectal valves, okay? And they make it more difficult for feces to leave. In the anal canal itself, you guys, we have the anal sinuses. These anal sinuses have a lot of mucus glands, which makes sense because you want to lubricate those feces as they're forced out of the anus. And if you look closely here, there's two anal sphincters. So there's an internal anal sphincter right here and an external anal sphincter out here. The internal anal sphincter is smooth muscle, and the external anal sphincter is skeletal muscle. So if the internal one's smooth muscle, is that under voluntary or involuntary control? Involuntary, right. Good. So that one's solely under the control of reflexes, like rectal reflexes. So once feces enter the rectum, then the internal anal sphincters would relax. But if that were the end of the story, that means you would actually defecate or poop every time feces entered your rectum. And that doesn't happen because obviously we can hold it, right? So how do we hold it? Well, we hold it because the external anal sphincter here is skeletal muscle, right? And skeletal muscle is under somatic motor control, which is conscious. And so what you can do then is actually squeeze the external anal sphincter. That way you don't defecate when it's not appropriate. But it takes time to learn how to use that, right? So it takes time to learn how to use that. And so little kids don't know how to use that yet, right? Obviously. And so that's why they can just defecate, even when they're not ready to defecate. And they know that that's not right because sometimes they feel guilty about it, but they don't know how to control it yet. They have to learn how to use that muscle, just like they have to learn how to use the muscles of their appendages. They have to learn how to use their external anal sphincters, okay? Now you guys see these veins here? These veins are called the hemorrhoidal veins, right? Or the superior and inferior rectal veins. But this is why they're called hemorrhoids. So what a hemorrhoid is an extrusion of these veins into the anus or into the rectum. Now that happens if there's too much pressure in these veins, okay? Now you get too much pressure in these veins, like if you're really straining to defecate, or if you have liver failure and blood's backing up into the veins of your portal system, right? Because these also drain towards your liver. So what a hemorrhoid would look like then would be like a cauliflower-shaped extrusion of venous material into the anal canal. So you can imagine then that could be painful because those are going to be pretty weak, and those hemorrhoids could potentially pop when you defecate, right? So that means that if you defecate, you could actually bleed a lot, and that's going to be painful. It's also going to put you at risk for infection because now you have a wound in your rectum where there's a lot of bacteria, okay? So that's why you can get hemorrhoid removal. It makes sense. Just have them lasered off or ligated off. There's a lot of different things, a lot of things you can do with hemorrhoids. But I think the key point, you guys, is just for most people, just avoid straining to defecate, And to avoid straining to defecate would be to prevent yourself from having constipation. And there's lots of things that can lead to constipation. It can be caused by medication. It can be caused by certain conditions like we've talked about already, like hypothyroidism, right? So but that's important to keep in mind. All right, so let's talk about the defecation. Actually, let's talk about bacterial flora. Just kidding. So with bacterial flora of your colon, you guys, flora would be like the population of bacteria, like flora and fauna. You guys heard that phrase? Now, it doesn't mean like the bouquet or like the flowers of your gut. I mean, you can look at it that way. Why not? In fact, if you think about the bacterial flora or the bouquet of bacteria in your large intestine, it would be a bouquet with lots of different types of flowers in there, right? Not one type of flower, like many different types, like a really complex bouquet. You're looking at the thing like, man, I can't even count how many flowers there are because there's just so many types, right? So the bacterial flora of your gut. Well, first of all, where do they come from? Well, they come from inoculation from your mother during labor. So if you came through the birth canal, right, instead of cesarean section, the first bacteria you're exposed to in significant quantities are the bacteria of the vaginal canal, and that actually colonizes your gut right then because, it gets on your mouth and then the bacteria make it down the whole way, right? So it also comes from just your environment, like living in a home with people that also have these bacterial flora. And as much as you'd like to think that we're like the cleanest things ever, we're not. Like our gut bacteria end up on items all the time because, when you defecate, they get sprayed out of the toilet in your bathroom, right? Or maybe people forget to wash their hands, they start touching things. So what's interesting then is that not only is your gut have all these bacteria, those bacteria that are in your gut are also in your environment. And you can get those bacteria from the people you're around as well, right? So what's also really cool, you guys, is that these bacterial flora, the ones you have, are dependent not only on your environment, but also your genetics. So for people that are very genetically similar to you, like very close relatives, they're going to have very similar gut flora as you, right? That's important to know. Now, their major function, though, of these bacterial flora is to colonize the colon, right? And by colonizing the colon, they occupy space. And by occupying space, they prevent bad bacteria from getting a nice foothold and kind of taking over, right? It's kind of like, nope, we're already here, can't colonize here. You know, they've already colonized that space. So that's pretty important. So it's involved with immune defense in that regard, right? Now, some of these flora also synthesize B vitamins as well as vitamin K, which is involved with blood clots. That's pretty important. B vitamins are involved with your metabolism. Okay, they help metabolize some host-derived molecules like mucin, heparin, and hyaluronic acid. But they also ferment indigestible carbohydrates. So when you guys hear about like fiber, that's indigestible carbohydrate. And so because you can't digest it, you don't absorb it, but your bacteria can digest it. So if you're eating foods that are high in fiber, those are good for your flora. But they also produce metabolites that from this digestion, right? And so usually the metabolites that are produced by these bacteria can be gaseous, right? Like hydrogen sulfide gas and carbon dioxide. That's what can lead to farting. So the composition of a fart would be the metabolic gases of your gut bacteria. It can also sometimes be a little bit of air that you swallow as well, right? So kind of interesting. Now they release these irritating acids and gases, and this says up to 500 milliliters a day. So if we go back to my water bottle example, right? It's half of that water bottle, 500 milliliters. That's kind of a lot of fart on average, right? It could be more, it could be less. It depends on what's going on. So it's kind of interesting. Cool. So take care of your flora, you guys. They're very important. So let's talk about Valsalva's maneuver, you guys. So Valsalva's maneuver would be the muscles of your rectum can contract to expel feces, right? Now, that's defecation, rather. Valsalva's maneuver is this. That's Valsalva's maneuver. So you guys remember how we just talked about the glottis in lab? And that was in your larynx. And we said that when the glottis closes, you go, huh, and it closes off. So the glottis assists in Valsalva's maneuver. But what you need are two major events. The closure of your glottis and the contraction of abdominal wall muscles, like your obliques and your rectus abdominis, and your transversus abdominis. So when those abdominal wall muscles contract, they increase pressure in the abdomen, right? But the reason why you want your glottis to close off is you don't want air to escape your lungs, because then the pressure would actually decrease inside your abdominal cavity. So what you need then is to close off your airway while you're contracting your abdominal muscles, and that's going to increase pressure in your abdominal cavity, which is the Valsalva's maneuver. All right, so what's that good for? Well, defecation, right? You guys know that sound, right? Okay, maybe not that intense. That's pretty dramatic. You're like, okay, but the closing of the glottis, right? Contraction of the diaphragm as well. So increasing abdominal pressure to try to force those feces out. However, there are also other pelvic floor muscles that are helping to elevate the rectum, and you have rectal contractions that are also going to force those feces out. But remember, you guys, you also need to have relaxation of the anal sphincters, because you can strain all you want, like, right? But if those anal sphincters are contracted, feces aren't going anywhere, okay? So how do you get the anal sphincters to relax? Well, there's two of them. You guys remember that? With the external and the internal anal sphincters. So if we look at this process here, we're looking at the reflexes for defecation that involve your rectum and anus. So what this slide is showing is our sigmoid colon, and then our rectum. So this is the distal end of your large intestine, and then here's the anal sphincters. So this is the internal anal sphincter and the external anal sphincter. What was the internal anal sphincter made of? Smooth muscle. Very good. And the external anal sphincter? Skeletal muscle. Nice. So here's the external anal sphincter, you guys. Let's follow the nerve fibers back up. Go do 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 do. Oh wait, here they go. They just came right from the spinal cord. In fact, those nerve fibers from the spinal cord are just somatic motor neurons that actually receive signals from your brain or your cerebral cortex. So the external anal sphincter then is a direct sort of conscious form of control from your brain and spinal cord. So what would you need to do then to relax that one? If it comes from your brain. So you're relaxing. It's not a complex answer, guys. It's actually pretty easy. Yeah. Just think about how. No, it's not a trick question. Yeah. What do you guys think? Yeah, you just kind of you think about it, right? You just kind of chill out, right? Like you start thinking, oh wait, I need to defecate. It's time to defecate now, right? Time to relax, and you just do it, right? I mean, none of us can really describe what that's like, other than we just know that that's what you do, right? But you have to learn how to do that. But that's your higher order brain centers, like your cerebral cortex, that's controlling the motor neurons of your spinal cord that can control the skeletal muscle. So you're basically going to prevent contraction of the external anal sphincter by just thinking it's time to defecate. But that's not the end of the story, though, right? Because we also have an internal anal sphincter. And what was the internal anal sphincter made of again? Smooth muscle. Nice. So that's under involuntary control. You have no conscious control over that. In fact, it relies on a reflex. So if you follow these nerve fibers back, well, they also come from the spinal cord. But do you guys see that in the spinal cord, these motor neurons, they receive their impulses from the rectum. Okay, let's think about this. If you have sensory fibers in the rectum that can communicate with motor neuron in the spinal cord that could potentially tell your sphincter to relax, what's going on here, you guys, is once feces enter the rectum here, because of stretch of the rectal wall, these sensory fibers send basically kind of like information to your spinal cord that inhibits these motor neurons from contracting the internal anal sphincter. Does that make sense? So if you inhibit the contraction of the muscle relaxes. But that's solely based on a reflex. However, do you guys see how it does involve your spinal cord? So what does this mean for people with spinal cord injury? They have a difficulty with defecation, for sure, because they can't necessarily rely. It depends on the type of spinal cord injury. But let's say if it's low enough to where it affects the sacral spinal nerves, then you're saying that they can't rely on the stretch reflex of the rectum to defecate. It's going to be much more difficult to defecate for them. Okay, good. But do you guys see how there's other pathways here that go out to muscle that's nearby? Like these nerve fibers can branch and they go out to muscle that's on the rectal wall and sigmoid colon. Now that actually causes contraction. So these nerve pathways that go to like the rectum and sigmoid colon, these actually cause contraction of the smooth muscle in those areas. That way you get simultaneous relaxation of the internal anal sphincter and contraction of the rectal wall. So that kind of makes sense, right? You guys ever experienced that before? You can like feel your rectum contracting? You don't have to admit to it. I definitely have. There's been times where I thought I wasn't going to make it. I'm like, oh no, I'm not going to make it. And you start getting sweaty. What's up with that? Why would you get sweat? Why would you sweat if you have to poop, but you don't really need to poop at that? Or it's not appropriate, right? Let's think about that. Sweating is what branch of your autonomic nervous system? Is that sympathetic or parasympathetic? Sympathetic, good. Your sympathetic nervous system inhibits defecation because defecation is a parasympathetic type of thing. It's a rest and digest type of response. So if you wanted to inhibit defecation, you have to become sympathetically active. So guess what? You start getting sweaty, your heart races, you're like, oh my God, I'm not going to make it, right? That for me, I was stuck in traffic. And I was like, oh my God, I was on I-25 and there's nowhere to pull over. I'm like, I'm done for. I'm done for. And I was on my way to go teach too. Good times. I did make it, by the way, which is good. So there's a happy ending to this story.